I guess just like connectome tracking algorithms depend on how you seed them. The driver's choice of route depends on how he's feeling in the morning. So yesterday he came by the main road and all was fine. Today he decided to go through some internal roads and it was like mayhem. Yeah, but anyway, so we'll get started. We'll go ahead with the theme of you know how do we use connectivity to understand something about behavior, right? So in that sense, today's talk might be quite different from many of the other talks you heard in the workshop, and and one of the main reasons I was planning to present this today is because especially for the folks who do a lot of modeling, I wanted you to also be exposed to behavioral models. So models that can allow you to understand components of behavior, and we'll see some of that in today's talk. Okay. So continuing on the theme of Varsha, this is also Varsha's part of her PhD work. So for those of you who were here yesterday, Varsha was a PhD student in the IMA program. Maybe I forgot to mention yesterday, but she was my first PhD student to graduate. So I got the maximum work out of her because I could give her a lot of attention. So yesterday she had one great paper, and today I'm going to talk about another great paper that she published. This was in PNAS, and this was about how we can use these types of connectivity correlates to measure attention's effects in the brain, right? So this is and these are the funding agencies. Okay, so we'll start with a simple experiment for you all to to get you all oriented to this idea of attention. Let's see this place. Yeah. So what I'm about to show you is two images, right? So this image will keep flashing, but there are really two images. They are alternating one after the other, and there's a major difference between the two images, right? It's not a small difference, a big difference between the two images. So if you spot the difference, I would like for you to raise your hand, right? Don't say it out, just raise your hand, okay? I guess this works slightly better in bigger halls because they, for them the image is much more within their field of view. So, okay, 1, 2, 3, okay, few more are seeing it. Okay, good. So for those of you who are still a little puzzled, if you notice there's a stack of books here, right? So I'm going to move my mouse cursor away. So there's a stack of books here, and this stack of books is appearing and disappearing, right? And if you if I directed your attention to that stack of books, you would of course be able to say that it's nobody then will say I don't see the change. But until I do that, many of you are a bit puzzled, right? Like it looks like a visual system can see this entire scene at high resolution, and yet so this let me just stop somewhere where you can actually see the scene. Yeah. So all of these books, all of these pillows, it looks like your visual system can take everything in, right? But really it looks like it can't, because even when there's a major change in the scene, interrupting that change or accompanying that change with a little gray blank throws your visual system off, right? It makes it very challenging to identify what is changing in the scene. This property is called change blindness, and one of the classic ways of testing attention in the lab, right? One of the many different ways we use to test attention in the lab. Okay, so what is attention? So this gives us a bit of a clue as to what we all in the community think of as attention. It is our ability to select the most important information at each moment in time for differential processing and decision making. So the idea is, in this room you have various kinds of information coming in at you. You may not even realize it, right? Because you're looking at this scene and there's there's this image on the screen, there's me talking, there's maybe other your friends moving around and fiddling with their phone, whatever it is, right? So but all of that you decide at each moment in time what is important, And why is that even relevant? That's because your sensorium, even though the senses that engage with the external world, they can take in a lot of information. Your brain can only process a small amount of information, as the change blindness demo showed you very clearly, right? Only a small portion gets access to higher cognitive functions, and that what decides what gets access, that's attention, right? So, the study of attention is not new. William James, over a century ago, defined attention as, he started out by saying everybody knows what attention is, right, thereby absolving himself of giving a formal definition, but then he went on to give a nice definition. He said it's the taking possession by the mind in clear and vivid form of one out of what seems several simultaneously possible objects or trains of thought, right. The idea is that there are many things, and then there's one thing that sort of engages your mind, as it were. Focalization and concentration of its essence, it implies withdrawal from some things in order to deal effectively with others, right. So, especially when there are irrelevant things in your environment, like perhaps some sounds or somebody walking into the door, etc., you would like to ignore them and then focus on things that are relevant, right. So, this is James' definition of attention. Again, just like yesterday, please feel free to interrupt me and ask questions, because this talk is going to, there's going to be lots of things. There's going to be behavior, there's going to be modeling, and then there's going to be connectivity, linking the behavior and the modeling. Okay, so what I'm going to tell you, some of this you may already be familiar with, but I'm going to perhaps introduce you to something that you're not very familiar with, right. So, if you consider attention as some kind of filter or bottleneck that decides what kind of sensory input is relevant for behavior, what kind of sensory input gains access to behavior, I will argue that there are at least two components to this process of selection of sensory input. One I'm going to call sensitivity, the other I'm going to call bias. So, these are terms that are used in the title, and I'm going to explain them now, right. So, what is sensitivity? Sensitivity is selection of relevant information for enhanced sensory processing. And what is bias? Bias is selection for prioritized decision making, right. So, there are two different kinds of things that are happening in the brain. One is enhancing the sensory processing of the stimulus itself. The other is deciding that something is more important than something else when we make decisions. Now, how do we do we have some intuitive examples of this in a day-to-day setting? So, imagine you're driving down a road, the freeway in this case, and there's a vehicle directly in front of you, right. So, you're the driver of the car behind. So, any driver who's careful would pay attention to this vehicle, because if that driver speeds up or slows down, you're going to have to react quickly, right. Whereas, if this truck slows down, you don't care too much because the truck's in another lane, right, and care least about this Jeep here, because really 2 lanes away, right, is less important. Vehicles on the other side of the median, even less important, right. They can speed up and slow down, they're all in your field of vision, but those things can speed up and slow down, but you don't care, right. So, whenever I give this example, this started out when I was a postdoc in the US, but then when I came to India, I realized it's not a great example, right. You have to react regardless of whichever of these guys moves, and then I also realized that the median doesn't matter too much, right. So, there can be a sudden break in the median when you least expect it, and the guy on the other side of the median can also come across. But nevertheless, we will assume that this is very disciplined traffic, and everybody is going to follow lane discipline, switch on indicators before turning and all that, right.
You didn't consider the pedestrian who decides to simply run across
that. Yeah, so you can see a conspicuous absence of humans in this picture, right. Nobody. Absolutely, pedestrian, cattle. So, but if you did, assuming that you did pay attention to this driver, I'm going to argue that one of two things can happen, right. One is you can enhance sensitivity to this object. Now, what does that mean? It means that this object becomes clearer in your perception. As many of you can probably see from where you're sitting, these objects have all become blurred, right, and this object becomes clearer. Maybe you can process the number plate of that car with a little bit more clarity, or fine movements of that car, right. So, you want to do that to be able to react quickly to them, right. So that is sensitivity, enhanced sensory fidelity, right? The other component of attention is bias. Now note that in this scene, the quality of the images of all the different objects in this scene and this scene are exactly the same, right? They look exactly the same. The only difference is that your brain has put in certain markers. It's decided that any information coming from this object is not going to be relevant for decision making. I'm not going to accelerate, slow down, turn, or anything based on what this guy does or what this guy does on the other side of the median, but I am going to be making decisions based on what this person does, right? Movements of this car. So this is filtering at a decisional level. You decide this is important, this is not important. These two processes, that is the enhanced sensory processing and enhanced decision making, generally go hand in hand, right? Attention typically, puts these two things in conjunction, so that wherever you are attending, that area becomes clearer in your perception, and you also generally tend to use that for decision making. But what we've shown, and I'll show you later today, is that in principle they can be separated, right? And in practice also I will show you how they can be separated. These are two distinct concepts. They are not the same thing, right? Is this idea clear of what is enhanced sensory processing and decision making? Okay. Yeah.
Practical is the enhancing sensory fidelity from an individual's perspective.
Practical meaning?
Like when you're sitting, the idea is sort of clear because you blurred the rest of the image, right? That doesn't happen in reality. You see everything.
No, that's exactly what I showed you with the change blindness demonstration, right? Your brain tricks you into believing you see everything. You actually don't see everything. Had you seen everything, you would have actually noticed that the stack of books was changing, right? The fact that you failed to notice that means that your brain this is all an illusion. The fact that you can see everything at once, and the major experiments have been done. So for example, if you're looking at this and I quietly replace this clock with a cuckoo clock or whatever, right? Completely different thing. People just fail to notice it because the word notice being the operationally important word, that's attention, right? So if that's because your visual system tricks you. I mean, you sample different parts of the scene in a very sparse way, and then your visual system reconstructs the entire scene. That's a trick your brain is playing on you. And if you change things in a way when people aren't paying attention, you get completely bamboozled, just like that change blindness demo I showed you. It's hard to believe, right? But that is the bottom line here.
Really nitpicking in the example you showed earlier for change blindness, there was a gray blacking screen. If that wouldn't be there, it would be fairly easy to.
So there's a slight nuance there which I didn't work on. So the point is, if you change the object if you make a big change when the person is looking at the screen, it activates your motion sensitive neurons. So we want to avoid activating your motion sensitive neurons, because that's a huge bottom up stimulus, right? That's like somebody walking in through the door. That makes it easy to see. But if I did it either gradually or did this little bit more of an abrupt manipulation where I washed out your iconic memory and then brought back the scene again, you wouldn't be able to perceive it. Or let's say there's somebody walking in and I changed this clock when some person walked in, you wouldn't be able to notice that I changed the clock when your attention is otherwise engaged, right? So there's a nuance there. We can discuss that. Okay, so so basically the idea is that there are these two essential components of attention. One is more sensory and one is more decisional, right? Okay, so I mean, are we the first to come up with this dichotomy or this distinction? Not really. If you notice, William James said in clear and vivid form, right? So that is already the idea of sensitivity, the idea that attention involves selection of information for enhanced clarity, and then he said withdrawal from some things in order to deal effectively with others. So that deal effectively is this idea of decision making. So that's the idea of bias. Yeah. So whether sensitivity and bias are really under the control of same or different mechanisms in the brain has been the subject of debate. Recently, actually started in the 80s, but work was trying to figure out whether these two substrates really are in the same regions of the brain. There is some evidence that they share overlapping substrates, and there's some evidence that they share they have distinct substrates in the brain, right? And this debate is really what today's talk is going to be about. Are they in the same areas of the brain, or are different areas responsible for controlling sensitivity and bias, right? Yeah, so but for that I'll have to introduce you to a little bit of behavior and modeling, and I wanted to again do this because many of you may not have. How many of you have done behavioral modeling here? I know many of you have done like neural modeling and so on, but anyways, anybody is familiar with signal detection theory perchance? No? Okay, good. So this may be a good way for you to get an idea of these theories, right? Okay, so this type of task is if you were doing an experiment on a human participant or an animal. Yesterday Collins was showing you the animal moving around in an environment, right? So that's a relatively free form of exploration. The animal is not necessarily performing any very strongly goal-directed task. It's sort of picking up the chocolate-covered cereal from the ground, nothing much more than that, right? But in very controlled settings, if you want to do experiments and look at things like attention, this is the type of task that you do, where you have the participant sitting on a rig. This can be a human, it can be a monkey. Nowadays, animals, rodents even do these types of tasks. So the animal is somehow immobilized in a rig, or the human is sitting with their head fixed in front of a visual screen, and there's a fixation dot here. They have to keep their eyes on the dot, and a target stimulus. In this case, this is like a little grating stimulus. That's a alternating pattern of dark and bright. That appears and it disappears. And what do you tell the participant or the animal to do? Is if you detected the stimulus that was briefly flashed, right? It's a dim stimulus that's briefly flashed, you make an eye movement. The eye movement is called a saccade. So you quickly make an eye movement and indicate to me that you've actually seen the stimulus. If you don't see a stimulus, right, if no stimulus is presented, you maintain fixation, don't move your eyes, right? So this is a very standard task in psychophysics literature. It's called a two alternative force choice task. 2 AFC or yes no task, right? So the participant either says yes, I saw a stimulus, this grating, or they say no, I didn't see a stimulus, right? Now we want to estimate things like sensitivity and bias from this using this type of task. Just one moment. Sensitivity for detecting the stimulus and bias for detecting the stimulus, and I'll tell you how that's done. Yeah.
So along with the response from the participant,
yeah,
is there a camera recording the saccade?
Yes, there will be some kind of eye tracking equipment that records where they made the eye movement, but the eye movement is slightly sort of less important here. They could be responding with the button press, etc. That's not that's not critical here. But with monkeys, for example, it's easier to train them to make eye movements rather than making them do very abstract things like pushing buttons and so on.
I misunderstood you, but so to check sensitivity and bias, my idea was that sensitivity is when the eye actually moves, and bias is when the participant thinks it's moving and they.
So good intuition, right? So these are things that one could sort of qualitatively hypothesize, but you want to quantify this, right? And that is exactly what we will do in a moment in a response modality independent fashion. So what I'm about to show you, something called a stimulus response table or a contingency table or a confusion matrix. Now those of you who do machine learning might be quite familiar with this, right? So this type of table is what you construct after your participant does hundreds of trials, right? Once they do hundreds of such trials, you construct such a table. The columns of this table are the two stimulus conditions, whether the target was actually present or whether it was absent, and the rows represent the response modality. So whether the participant said yes or participant said no. So if the participant said yes, I saw a target when it was actually presented, that's called a hit, right? And if the target was absent and the participant said no, I didn't see a target, that's called a correct rejection, right? So these two are essentially the correct response types. If these correct response types are more and more, right, if they increase, that means that your sensitivity is higher, you're actually able to tell apart the presence of the target from its absence. But there are other types of errors that the participant can make. So sometimes when there is no target presented, the participant can say, yes, I saw a target, right. So that happens quite frequently, especially when the target is dim or it is displayed in a background of noise, right. So then it becomes actually difficult to tell whether the target was presented or not, and then sometimes the participant says, yes, I saw something, even though actually nothing was presented. And sometimes when the target is dim, again the target is presented, but the participant says no, that's called a miss, right. So these two are incorrect response types and these two are the correct response types. Yeah. So this is a contingency table that you'll see very often in two alternative force choice or various kinds of behavioral tasks. Yeah. Okay, so now how do we estimate sensitivity and bias from this? Like somebody was saying, the idea that you can perceive the target better, that is measured with this index called D prime. Now the way you do this is using a framework called signal detection theory, and to actually explain signal detection theory it will take a while, right. So I will give you like a 30-second summary of what signal detection theory is. Now, what signal detection theory says is you have a decision variable that's an internal variable, a latent variable that's distributed as a Gaussian, like a normal distribution, and that distribution has a certain mean, right, when no target is presented. So that's this noise distribution here. But when you actually present a target, the whole distribution shifts rightward, right, and the mean value of the decision variable increases, and that is this red distribution here, right. That's when the target is presented. The distance between these two distributions, the means of these two distributions, the separation, that's called D prime. That's the sensitivity index, and that quantifies the discriminability of signal from noise, or basically the idea that you can actually detect the target versus no target, right. And the way you compute D prime is using a formula. I won't go into the details of the formula. This function phi inverse, does anybody know what phi inverse is? We've seen this function before. No, it's called a probit function. It is the inverse of the CDF of the normal distribution, right. Yeah, so this phi inverse is a probit function, and you just take the difference of the hit rates minus the false alarm rates to give you this index of sensitivity. So let's get some intuitions for this. Yeah.
In that graph, what does it mean by the intersection point? Like the red and the blue, right? So the intersection point, what does it mean? Like that thing is shown to it, or?
So what happens? So this is the distribution of the decision variable over many trials, right? So if that decision variable value occurs exactly at the intersection point, then your guess is as good as anybody else's, right? Because then your brain won't be able to tell whether it was actually shown or not, right? So if you put a threshold value here, and any time the decision variable is greater than the threshold value, you say yes, and when it's less than this value, you say no. That's how a rational observer would make decisions, right? But if it falls right at that intersection point, it's it's just chance. You can't tell whether it was shown or not, right?
You can create an error.
Sorry?
You can create an error.
It could create an error, but it could also equally likely create a correct response, right? So at that point, you're sort of breaking even. Right? Okay, so let's get some intuition for this formula here. I'm claiming that sensitivity is something related to hit rates and false alarm rates. So let's go back to the contingency table and look at why I'm looking at, in some sense, a difference between H minus F, right? So I'm looking at this difference between hit rates and false alarm rates. Hit rates intuitive, right? So if I have a higher hit rate, that means I'm likely to have a higher sensitivity because this is the correct response. But why am I subtracting out the false alarm rates? Why is that happening to estimate sensitivity? Any thoughts? Why should I subtract out the proportion of this type of incorrect response? So under what circumstances can I get a very large hit rate? Is it only if I see the target well? You can keep pressing yes, right? So if I say yes to everything, then I'll have a very high hit rate, regardless of whether I see the target or I don't see the target, whether the target is actually presented or not, I can get a large hit rate, but I will also get a large false alarm rate in that case. And so then you have to sort of balance these two things out. So only if you have a high hit rate and a correspondingly low false alarm rate, only then do you have real evidence that the participant is discriminating the presence of a target from its absence, right. So that's the intuition, right. So to be fair, I cover this in about 3 months of a signal detection theory class. So I'm going through it in 30 seconds. So if you don't fully get it, no problem, right. So we'll cover it in more detail offline. So that was sensitivity. Now how do you estimate bias, right. So for bias, we define this cutoff value that we call or a threshold that we call a criterion, right. Like somebody mentioned here, the optimal criterion is when you put that threshold value right between the red and the blue distributions, right. So to put it in sort of mathematical terms, these distributions, if you think of them as likelihoods, this criterion value right at the intersection is the point of equal likelihood, right. Equal likelihood of observing the target under a certain value of the decision variable, right. So you can write these as conditional probabilities, likelihoods. There are many different ways to formulate this. So if the decision variable value on a given trial exceeds this criterion, the participant says yes, and if it falls below the criterion, the participant says no. But participants don't have to always act as rational observers, right. So not everybody will put the criterion exactly between the red and the blue distributions. Some of them will put it all the way to the left, and some of them will put it all the way to the right. So if the participant puts the criterion all the way to the left, which type of response is more likely? Yes or no? The yes response is a lot more likely, right, because if you put this criterion all the way to the left, then pretty much any value of the decision variable here along this axis will prompt you to say yes, because your decision variable is going to be higher than the criterion, right. And just one moment, so that's called a liberal bias. Now if you move the criterion all the way to the right, you need lots of evidence before you say yes, right. So most often the participant will say no, I didn't see anything. That's called a conservative criterion, right. And just one moment, so there's a way to measure this, and that is using the sum of the hit rates and false alarm rates, and this C, or sometimes we'll call this BCC, is the criterion value that allows you to tell whether the participant was liberal or conservative, right, where they're more they have a preponderance to say yes more often, yes I saw something, or did they have a preponderance to say no more often, right, I did not see something. Yeah, what was the question?
Did you explain how we are going to calculate the distribution?
Yes, so I will, there's again a nuance that I didn't get into. The distribution is a latent distribution, it is not directly observed. So what signal detection theory posits is that there are these two distributions that are built inside your brain. Very loosely speaking, you can think of them as distributions of some neurons firing rates, right. So when no target is presented, the neuron fires on each trial a certain number of spikes that is drawn from this distribution. On trials where the target is presented, it fires a certain number of spikes that is drawn from this distribution. And I am going to posit that these are Gaussians and that they are only mean shifted, right. There is no other change in the, there is no difference in the standard deviation, etc. So for those of you who would actually done neural modeling, why is this a terrible assumption to treat this as a neural firing rate? What's the sort of distribution that neural firing rates follow? Mathematical distribution that neural firing rates follow? Poisson, right? So neural firing rates follow a Poisson distribution. So this is, I'm only saying neural firing rates to give you some handle on what this is, right? It's not neural firing rate, something else. But think of it as some kind of population activity for the moment, right? So the neural population activity can be described by this distribution when the target is not presented, by this distribution when the target is presented. And how do you construct these distributions? You don't. You assume they exist. You only characterize the parameters of these two distributions, that is the distance between their means, as a fraction of the standard deviation. that is d prime, and criterion is really given by this formula, which is where this threshold sits relative to the intersection point of these two distributions. So, if it sits to the left, you have a liberal bias. If it sits all the way to the right, you have a conservative bias, right. You do not actually characterize the distributions themselves directly. It is like a latent variable model, right. So, you only get the parameters indirectly, the parameters of the distribution. You do not directly measure the distributions themselves. You could, I mean, if you actually did measurements in the brain, you could, and people have done it, but we will not be going.
Cap on people's head using like a cap with a bunch of electrodes.
For measuring these distributions?
Yeah.
Even with that, it will be very challenging. To actually measure these distributions, you need to record neural activity across many different brain areas with lots of fine-grained resolution, right. So, with EEG, what you are describing, measuring the distributions will not be straightforward. Okay. But this is the nice thing with behavior, right. You do not need any caps. All you need is the stimulus response contingency table based on participants' responses, and you infer d prime and criterion. And it turns out that if you make predictions of people's behaviors based on these parameter estimates, they are pretty good, right. So, there is something accurate about this model which describes how people make decisions. So, this signal detection theory is to my knowledge in the last 20 years plus in neuroscience, one of the most well validated theories of behavior in in neuroscience. Very few theories are as well validated as signal for behavior, right, for behaving, capturing aspects of behavior. Okay, so it looks like we have a way to measure sensitivity and criterion. So, what is the challenge? What is our problem here? It turns out that especially in attention tasks and the tasks that they use in monkeys and also in humans, there is a third alternative. Everything I showed you so far was a two alternative force choice task, yes or no, right, left or right and so on. Those are all two alternative force choice tasks. So, what is a common example of a three alternative task? So, this is something that you may have seen as a two alternative force choice task, right. So, there is a choice between Democrats or Republicans, but then you have candidates who say either immigrants are eating our pets, or I mean, he has more interesting things he said since I made this slide, right. So, you have got these two extremes that neither of which you want to support. Then the Indian government at least gives you an option, which is nota. You can say none of the above, right. There is a third option, which is to say I do not want X, I do not want Y, I am going to say neither, right. And in the US also, people potentially exercise this option implicitly by not voting in the polls. There is only about 60-65% of the people vote, right. So, most of the time they do not. So, at least not most of the time, but a significant fraction of them do not. So, the idea here is that there are sometimes implicitly or explicitly three alternatives, right. So, there is not many real life tasks are not two alternative tasks, and these types of tasks we are going to call multi alternative tasks. The moment you go more than 2, n greater than 2, we are going to call them multi alternative tasks. The type of tasks I told you are detection tasks, because you tell whether the target was presented or not, yes or no. So, therefore, they are multi alternative detection tasks. Now, how exactly does such a task go? So, you got two locations on the screen where the target can appear. Previously, there was only one location, remember. So, in this case, the participant or the animal makes an eye movement to one of the two locations depending on where the animal perceived the target. On some trials, there is no target and the subject maintains fixation, right. So, they do not move their eyes to either location, they just maintain fixation. And you can easily see that you can expand this to any number of locations, 3, 4, 5, 6, etc. The simplest version of this task has two locations. Now, why do you need two locations? At least because if you are studying a process like attention, you need one location that is attended, and as a control, you need another location that is unattended, right. So, if you want to study how your performance changes at the attended location relative to the unattended location, you need at least two locations, maybe more, and many tasks of this type are all multi alternative that have been used in literature for studying attention are all tasks of this type, right. And typically what these we do when we present tasks of this type is we cue the participant to pay attention. We actually tell them, on this trial you are going to pay attention to location 1, on the next trial you are going to pay attention to location 2, and so on. So in every trial the participant knows where they should be paying attention, and we measure their behavior at the attended location versus the unattended location to understand both the behavioral consequences and the neural consequences of attention, right. Okay, so now comes the question which has not been addressed surprisingly for many decades. How do you estimate sensitivity and bias in a task like this? The moment I show you this table, you would probably guess why this is challenging, because it's no longer a 2x2 stimulus response contingency table, right. So there's a cued location, that is the attended location, the unattended location, and there are some trials where the target does not appear at all. So the target may appear here, it may appear there, or it may not appear at all. The participant is also free to give any of the 3 types of responses. They could say, I saw a target at the attended location, at the unattended location, or I did not see the target at all. So that gives you 9 combinations, right. Suddenly there's an explosion in the number of combinations that you can model. And so unlike just the 4 stimulus response contingencies before, now there are 2 types of hits, 2 types of false alarms, 2 types of misses, and 2 other very strange responses which, believe it or not, even human participants give. You present the target at one location and you present it briefly enough, they will mislocalize it. They will actually say, I saw it, but I saw it at the other location, right. And this type of mislocalization response is also possible, and it does occur even in practice when you run tasks of this type, right. So now how do you get sensitivity and bias in a task like this? I won't belabor you with the details because there's a fair amount of math behind this, but I will just say that you have to extend signal detection theory to multiple dimensions. The extension is pretty straightforward, the math is a little bit hairy, but you can do it if you work through this very carefully. And the idea is very simply that instead of a 1-dimensional signal and noise distribution, now you have 2-dimensional signal and noise distribution. So along each axis, right, each of these 2 axes, you have evidence for the target at location 1 and the target at location 2, and your decision variable distribution is now a bivariate distribution. There before it was a univariate Gaussian, now it's a bivariate Gaussian, right. So when there is neither signal nor target, this distribution is centered around the origin, this bivariate distribution, and then when you present the target at one or the other location, it sort of translates like this, right. So it moves along either one axis or it moves along the other axis. So that's essentially the intuition for how a 1-dimensional model gets generalized to a 2-dimensional model. The trickiest part of this, again I won't go into this in the interest of time, there are many references that you can look at if you want to understand this type of model. The trickiest part is modeling decisions, right. Before, what we did is we had one criterion and we just said, okay, I'm going to put the criterion here versus there, and if the decision variable exceeds the criterion, I'm going to say yes versus no. This is a little bit more complex because you have 2-dimensional perceptual space and you have to make one of 3 choices. You have to say, I saw the target at location 1, location 2, or not at all, and therefore your decision surface now looks like a Y-shaped boundary, right. So and this boundary, depending on the value of the criteria, can translate freely on this 2-dimensional plane. It can't rotate, but it can translate, right. It can move around freely on this 3-dimensional plane, on this 2-dimensional plane, and using the 9 stimulus response contingencies that we capture in the stimulus response contingency table, we fit 2 sensitivities and 2 criterias, one pair of D prime and C for each location. For the cued location, we'll get a sensitivity and criterion, and for the uncued location, we'll get a sensitivity and a criterion, right. So this is the basic outline of how you capture sensitivity and criterion in multi-alternative detection tasks, right. Yeah. So, okay. So now we are ready to now use all this power of signal detection theory and answer some questions about where the sensitivity and bias are implemented in the brain. So, I want to start with the preemptive apology on terminology, right. I always give this apology because for people not used to signal detection theory, it can throw them off. I told you about two indices, right, D prime and criterion. A higher D prime always means higher sensitivity. So, there we are safe, right, no problem. A lower criterion, or whenever I use this terminology, a lower CC, it means a higher bias. Why? Because a lower criterion means you are more readily going to say yes, right, which means you are more biased to saying yes, more biased to selecting that location. So, this inverse relationship will plague us throughout the rest of the talk, right, and I want you to be a little bit alert, and I will point you to when signs are getting flipped and so on. So, lower criterion means a higher bias, a higher D prime, no problem, it means a higher sensitivity. Yeah. Okay, so maybe take a quick pause here. Any questions so far? I know there's a lot of information to digest, but I hope you got things at least at a high level as to where we've gone. Any so all we've done is introduced you to a multi-dimensional signal detection framework that's going to allow you to estimate sensitivity and bias, right. So, the two components of attention, right. Okay, so what can we learn about attention in the brain based on this? Yeah. So, for those of you who have not seen this, this is a side view of the brain, but after taking a section, right, a schematic of a section of the brain through the middle. And this area, does anybody know which way the eye is oriented? This is the eye, right. So, the eye is here, and all of the information from the eye reaches the back of the brain, surprisingly, right. So, all the visual information processing is happening here in the back of the brain. What is this area called? Have you all heard of this? Occipital cortex, right. So, this area is the occipital cortex. This area is called the parietal cortex, the area just above the occipital. So, back of the eye is the occipital cortex. Sometimes when somebody hits the back of your eye, you see stars, back of the head you see stars, right. That's because these neurons are getting mechanically driven, right. So, that's literally why the information, we know that information goes to the back of the head, back of the brain. This area just above it is called the parietal cortex, right. So, this area just above, just here, this is called the parietal cortex. This area is the prefrontal cortex, the area that is in the front, right. That is the prefrontal cortex. This is one of the evolutionarily most recent areas in the brain, right. And then there are other parts of the brain, and these are all thought to be important for attention. The prefrontal cortex, parietal cortex, striatum, and so on. And there is one particular area that we are going to look at that I have been looking at for a while now. There is an area here nestled up here called the superior colliculus. Now, that area is important for eye movements. We know that because if you pass small amounts of current into the superior colliculus of animals, they start to move their eyes, right. So, they start moving their eyes to whichever neuron you are tracking. So, that area which controls eye movements, which is in the brain stem nestled deep within the brain, is called the superior colliculus, right. And these areas, the prefrontal, parietal cortex and all, are evolutionarily fairly recent. Humans have a very advanced prefrontal cortex. Animals do not have a very advanced strong prefrontal cortex, but the superior colliculus is an area that has been in various species, starting from fish to birds to turtles. All of them have it. It is called the optic tectum in those species, but humans also have it. And one of the sort of major questions in the field has been, what is the role of the superior colliculus in attention? Like, does the superior colliculus actually play a role in attention, right? Okay, so what do we know about the superior colliculus? It is an evolutionarily conserved structure. It is in the midbrain, right? It projects to parts of the brain that drive eye movements. So, if you pass currents into the colliculus, it will move your eyes. You will end up moving your eyes involuntarily, right? So, you just have to stimulate it and your eyes will move. And there is some recent evidence suggesting that it is important for controlling attention, right. So, what is that evidence? That is what we will review in a moment, right. Okay. So, we will review this particular experiment that was done by Zenon and Krauslis. It was published in Nature a few years ago now. This why was this experiment published in Nature? Because it was a heroic experiment where they targeted this tiny midbrain structure and went in and inactivated it. So many of you were asking questions to Collins yesterday about what happens if you inactivate grid cells and so on. Those are probably challenging experiments, but this is an even more challenging experiment because the superior colliculus. So what's in the brainstem? What are some of the structures in the brainstem? Do we know? What if you went and tried to shut down some neurons in the brainstem? What would happen? Sorry?
Stroke.
There are, what do neurons in the brainstem control? They control breathing, they control various vital body functions, right? So if you accidentally shut down some neurons that or descending connections that are adjacent to the brainstem, the area that you're targeting, the animal will pass away, right? So you have to be very, very careful in terms of how you are targeting and ensure that you're targeting only the superior colliculus and not the other parts of the brain. Superior colliculus incidentally receives information directly from the retina and also receives auditory information, right? So it's very well poised to drive gaze and eye movements. Okay. So what these guys did is they went and inactivated the superior colliculus when the animal was performing an attention task, right? So this type of task here, what is this task? So these animals see a patch of dots, and these dots are moving in one direction or another direction, and what the animal has to do is whenever the dots slightly change in their direction of motion, right, when things are moving around on the screen and when that motion direction changes, the animal has to push a button. So they were sort of savvy enough to train monkeys to push buttons. It's not an easy task, right, because monkeys pushing buttons, they're not used to pushing buttons, right. So unlike us, constantly pushing buttons on the phone, the monkeys are not trained for this, right. So you had to train them for months, sometimes years on end, and then you get tasks like this. But what the animal has to do importantly is to ignore any changes that happen in an irrelevant patch. So that's how they're studying attention, right. So the monkey has to attend to this patch, push button only when this patch changes in direction. Anything happens in this patch, completely ignore it, right. You should not pay attention to it. This is called a target and this is called a foil, right. So what? Sorry?
Between two populations, those will be same for them.
The animal is cued. If you notice, initially there's a cluster of dots that's presented. So the animal knows that on this trial, I have to pay attention to things on the lower left and ignore things on the upper right. On some other trials, this patch will come up there. So on those trials, the animal knows to pay attention there and ignore things here.
Whichever comes first.
Correct. Whichever, this is the cue. This cluster of dots is the cue. This is telling the animal where to pay attention. Right? Yeah, that's a good question. Okay, so then they went in and inactivated the colliculus and they said, okay, what happens to the animal, right? So what they observed is that we've been discussing this hit rate and false alarm rate, right, as things in signal detection theory. They saw a strong decrease in hit rates when the superior colliculus was inactivated, and they also saw an increase in false alarm rates, right. So hit rates decreased, false alarm rates increased, and this was a surprising and powerful result because it showed that the superior colliculus is actually important for attention. It decreases, if you inactivate it, your hit rates go down, your false alarm rates go up. Now, if you've been following along with the signal detection theory that I showed you before, if hit rates decrease and false alarm rates increase, what happens to sensitivity? It would reduce, right? That's the conclusion, because I told you it's related to the difference in some way of the hit rates minus the false alarm rates. So if I reduce my hit rates, increase my false alarm rates, my sensitivity changes, right?
How do they deactivate superior falling?
They use a certain drug that goes in and called mucimol. So you go in and you silence the neurons transiently. Okay, so it produces detection deficits during an attention task, and like you all have mentioned, they interpreted this as a change in sensitivity. But it turns out that the story doesn't stop there, because the intuitions that you have about sensitivity and bias in one-dimensional signal detection theory get completely appended if you go into multidimensional signal detection theory. And what we were able to show is that without changing the sensitivity at all, only by changing the bias, right, only by taking this Y-shaped decision surface and moving it around, you can get exactly the same pattern of decrease in hit rates and increase in false alarm rates. But you can do this only in multiple dimensions. You won't get this result if you do it with one dimensional signal detection theory, right. So then the question is still open. What they observed, was it a change in sensitivity or a change in bias? So we took their data, we analyzed it, and that's part of that paper, and our results were more consistent with a change in bias, not sensitivity, right. So now Varsha came along, and this is when I moved from my postdoc to my independent position, and I was like dead set on figuring out what role does the superior colliculus play in humans. Is it sensitivity or is it bias? And so Varsha started this very nice line of diffusion MRI experiments. She wasn't particularly interested in behavior, but I said, hey, if you're doing diffusion MRI, also learn and do behavior on the side. And so this is the task that she designed, very simple and very similar to what we showed before. So instead of having two moving dots, right, you have two gratings, instead of that, you have two sort of gratings, alternating patterns of black and white, and you cue the participant on each trial to attend to one side or the other, right, same as the monkey task, very similar, right. Then after a certain window, these two gratings, any one of them could change in orientation. So in this case, you see the cued grating has tilted off, right, it's gone off to one side, and what the participant has to say, again, it's a multi-alternative detection task. Did the change happen on the right, did it happen on the left, or not at all, right? Did these two gratings stay the same, right, before and after this blank? So this is exactly similar to what the task that the monkeys were doing, except that we're using a different kind of stimulus than a moving dot display, right. And so she analyzed all of this with the MADC model and the multidimensional signal detection theory model. So what did she find? So let me cut a long story short. What she found is conventional signatures of sensitivity and bias. So that we verified, right. So wherever the animal was paying attention, sensitivity was higher and criterion was lower, which means bias was higher, right. This inverse relationship is what I told you will get confusing occasionally. So sensitivity was higher at the cued location, but bias was also higher, right. So this BCC more negative means higher bias, yeah. But what she also found, and this was actually one of the first reports from the lab, and we've replicated it since in many other data sets, there was an asymmetry in the sensitivity. Now that people had seen before. There was higher D prime for left side targets compared to right side targets. You tend to focus your D prime more on the left than the right. If you look at the visual world, people tend to be a little bit more sensitive to information on the left than the right. But bias actually goes the other way around. People tended to be more biased for information on the right than the left. So whenever they responded more frequently to changes on the right than the left. They were more conservative on the left and more liberal on the right, right. So this was again a pattern that again we've been, this was a study with n equals 22, but now we have n of over 100 and we've replicated this fairly thoroughly, right. So this type of asymmetry, visual field asymmetry in sensitivity and bias is something that participants innately have, right. This is all humans. This is all humans, and no, we don't work with animals at all in the lab. It's all human work. So now, if you want to look inside the brain, yeah.
Is this only for right-handed people or left-handed people?
So left-handed people were not included, but the mapping did not include a bias in terms of the motor response. So we actually made them respond up versus down, and there was also a cohort, because the reviewers asked us all the same questions, right? So we also included a cohort where the mapping was completely randomized. They could not tell which side they had to respond for each type of stimulus, right? So we were.
Since you were talking about this, you were relating to some innate bias.
Correct.
So that the handedness may be playing
I'll show you that in just a second, why we don't believe that's the case. So again, the mappings were randomized. So we ensured that it was not a low level motoric bias, right? So that level of control we have. But if you're interested, I'm happy to talk about it in more detail, because the reviewers asked us exactly the same questions. Was it handedness and so on?
Is it in non-human primates? This is reported already, right? This asymmetry in the
D prime is reported,
in D prime.
but bias,
But this is not a multi-dimensional signal detection.
presumably, right? So we don't know. Correct.
That's interesting because like monkeys typically are ambidextrous. I mean, they are not like knowing at least
Yeah,
in terms of motor
absolutely.
code.
That is the reason why I was thinking about it.
So, but those experiments would be interesting too. This is all humans. Everything that we've done is in humans. Okay, so then what can we do? Because we couldn't directly go in and do the same sorts of measurements that Krausless and others did, because you can't go into a human and measure from their superior colliculus. What Varsha did is manually delineate the superior colliculus and measure connectivity from the superior colliculus to all the different cortical areas. And some of the streamlines, just to give you an idea, this is what they look like, right? So yesterday, if you remember, I showed you some techniques to estimate streamlines. And so if you map from this subcortical area to various regions of the cortex, these are the types of streamlines that we get, these types of connections of fiber bundles, right? So what did she see when she actually did this? Now you actually see a asymmetry in the way the superior colliculus connects to the rest of the cortex. So it doesn't connect in the same way. There's a very strong left hemispheric bias. So the superior colliculus connects more strongly to left hemispheric regions compared to right hemispheric regions, and we analyze this two different ways. One is to look at the asymmetry in connectivity across regions for particular participants, and the other way is to average across regions and look across participants. In both cases, we saw that predominantly there were left-sided asymmetries in the connectivity. Now, the next thing that we did is to try to replicate this, because if you show in a sample like this, the reviewers will say, oh, that's because your scanning was biased, right? So we tried to replicate this in a cohort of participants from the Human Connectome Project database. So many of you may have heard of this HCP database that has scans, and here what we did, exactly like Arpan was pointing out, is to avoid this bias of left and right handers. We selected an equal proportion of left and right handers, because it's very hard to get left handers data, but HCP actually has 1,200 participants, right? So you have a free sample of left handers data, and so we selected the first 30 right handers and the first 30 left handers from that cohort, and we did this analysis, and we tried to replicate these asymmetries again in this cohort as well. So behavior is asymmetric, with bias being stronger on the right than the left, and superior colliculus connectivity is asymmetric, with connections that are stronger on the left than the right. Okay, so now you can guess where this story is going, and this is the last data slide. Then what we asked is, for a given individual, can we predict the asymmetry in their behavior based on the asymmetry in the superior colliculus connectivity? And for that, we have a linear regression model. The details are, I'm going to skip them. But basically what you're doing is you're taking a feature vector of the connectivity matrix, which we saw something like this yesterday. And for each participant, you predict the level of behavioral asymmetry, right? And you do that, using a leave one out or leave k out approach, a k-fold validation approach. And what we found is that the superior colliculus connectivity asymmetry could not predict asymmetries in sensitivity, but it could strongly predict asymmetries in bias, right? And Varsha has done many different control analysis. This being PNAS, he wouldn't let us off with one or two. So we have a supplementary section that is 3 times as long as the main paper itself. And so there we showed that this is specific to the superior colliculus, that that if you look at areas like the visual cortex, those connections are more correlated with sensitivity than with bias, that these asymmetries are robust, and so on and so forth. But to cut a long story short, this type of regression and prediction analysis allowed us to make a very specific connection between asymmetry in connectivity in the superior colliculus and asymmetries in behavior, and specifically the bias component of attention, right? Okay, so it predicted asymmetries in bias, but not asymmetry in sensitivity. Yeah. Okay, so I'm going to quickly summarize. So I've taken you through a very long sort of odyssey of lots of background information. I realize only some of which may have filtered through, but this I mean, I'm also happy to talk to you afterwards if you're interested in learning more about these things. But these are the key points, right? So whenever somebody says, "Are you paying attention?" you should remember that that is not a single construct that they're talking about, right? We're increasingly realizing in our field that attention is really an overloaded term. There's many subparts and components to it, and there are at least two that I've told you about today: sensitivity and bias, and there are other parts to it as well, right? And are these all governed by the same neural bases, or are there different parts of the brain controlling it? This is a question that's driven us for a while now. Our recent analysis suggested that there are very strong hemifield asymmetries in sensitivity and bias, and this connectivity analysis, the thing that was enabled by all of the methods that I spoke about yesterday, showed us that there's also connectivity asymmetries, particularly for this midbrain structure, the superior colliculus. And then what we were able to show is a specific sort of intersection between asymmetries in superior colliculus connectivity and asymmetries in one particular component of attention. And so this suggests to us, like taking the story forward, that the superior colliculus plays a putative role in modulating one particular component of attention, and it sort of brings that bigger debate, I think, to a advances that debate a little bit. I mean, scientific debates never come to a close, right? So it advances the debate a little bit, and so hopefully this type of intersection of neural and behavioral modeling is what will allow us to better and better tease apart, right, how the brain produces various aspects of behavior. And one of the reasons why we study only humans in the lab is because of this reason, that we can train them to do very exquisite behaviors very easily, right? So within for a monkey to make the saccade and all that Rich Krauselis did, push a button, it would have taken them months. For us to train a human on a task of equivalent complexity takes us 1 day, right? And so that's why we do all of this behavior in the lab, and also why we measure brain signals with non-invasive approaches. Okay, so thank you all for your attention, and happy to discuss with you during the break.
Two questions.
Yeah.
One, do you see at later studies, as you say, after these, not just the asymmetry in the behavior, but also functional asymmetry?
Yes.
In terms of neuroimaging data or things like that.
That's a good question. So we haven't looked at it very carefully in terms of like whether fMRI networks show these types of lateralities, but that would be an interesting thing to do. So the lab does a fair amount of like machine learning with fMRI, but we haven't probed the asymmetry question that carefully.
Went down all the way to connectivity asymmetry.
Correct.
At the structural level.
Correct.
I was wondering whether this is also present in
the functional level. That's a good question. There's something interesting to that.
Second thing I was thinking, could this asymmetry, actually the connectivity, that how superior colliculus connects to rest of the brain areas,
right,
whether it is right or the left, cortex, is mainly that it is choosing a shortest path length, or is it something to do with the diffusion actually process?
Shortest path length meaning at the axons themselves?
Right, exactly. Because in some cases, in auditory asymmetry, for example,
right,
it has been shown that between right and left auditory cortex, it is embedded. Right auditory cortex is more closer embedded, but not because the shortest path lengths that you measure from graph theoretical measures,
right,
being different between right and left auditory cortex, but it is because of the diffusion or any communication measures of diffusion being different between the right and the left auditory cortex when it kind of communicates with other brain areas.
So by diffusion, you're talking about diffusion of information.
Right.
I see.
Like spread of diffusion or communicability or those kind of measures, right?
Sure.
Like used by Bratislav Mizik and other kind of people.
Sure. I mean, I think that's definitely possible. Here,
From the anatomical connectivity very well.
correct. So here, I'm not sure we quantified the path lengths of the fibers. So that's something that we could do, because we actually have the connectome. So we can actually quantify the path lengths of the fibers and see if there's a difference there. Here we only thought of, we counted the density of the connections.